Unlike eukaryotes, prokaryotes have circular DNA that follows the theta model. From the outside it looks like this, with the red area being the replication bubble and our origin of replication. Let's look a little closer at this area. We start off with double-stranded DNA that is connected through the hydrogen bonds between the nucleotides. These hydrogen bonds are then broken with the helicase enzyme to unwind the DNA into two single strands. As the DNA unwinds, two other enzymes attach to the single strands. Single-stranded binding proteins latch onto parts of the strands close to the replication fork to prevent DNA from winding back up. Then gyrase also attaches itself close to the fork so that the tension is relieved as the DNA unwinds. This is done by breaking and reforming the phosphodiester bonds located between the phosphate group and the sugar base on DNA. Since DNA can only synthesize in a 5 to 3' direction, the leading strand will be the top strand. The old DNA will be red and a new strand will form from left to right, while the bottom strand is the lagging strand. Being complementary to the top, a new strand will form from right to left. Another enzyme known as RNA primase synthesizes primers which latch onto the DNA to begin replication. DNA polymerase III then attaches itself to the same location as the primer to read the DNA and begin adding nucleotides to form the new strand. On the leading strand, only one primer is required to extend the strand, forming a continuous chain. However, on the lagging strand, the strand is extended away from the replication fork. This causes fragments known as Okazaki fragments to form rather than a continuous chain. In this case, a primer is needed for each fragment. When the new strands are formed, DNA polymerase I comes in to remove the primers and fill in the missing gaps of the Okazaki fragments. These fragments are then properly sealed with the enzyme ligase. In addition to filling the gaps of DNA polymerase I also has an exonuclease function in which it proofreads the DNA to check for errors during replication. If an error is found, it can replace the nucleotides with the correct sequence. If it can no longer perform this function, the DNA will not be read for errors and mistakes, and this will increase the likelihood of mutations occurring in DNA. These are my sources, and thank you for watching.